Hello everyone, this is Priyanka Gupta from Dr. Hari Singh Gaur Vishwavidyalaya, a research scholar in Department of Zoology. In this video, I am going to explain bioinformatics protein sequencing databases. First of all, we should know about what are the biological databases. So, biological databases are libraries of biological sciences collected from scientific experiments, published literatures, high throughput experiment technology, and computational analysis. They contain information from research areas including genomics, proteomics, metabolomics, microarray, gene expression, and phylogenetics. Information contained in biological databases includes gene function, structure, localization, both cellular and chromosomal, clinical effects of mutations, as well as similarities of biological sequences and structures. Biological databases can be broadly classified into sequence, structure, and functional databases. Nucleic acid and protein sequences are stored in sequence databases, and structure databases store solved structures of RNA and proteins. Functional databases provide information on the physiological role of gene products, for example, enzyme activities, mutant phenotypes, or biological pathways. Model organism databases are functional databases that provide species-specific data. Databases are important tools in assisting scientists to analyze and explain a host of biological phenomena, from the structure of biomolecules and their interactions to the whole metabolism of organisms and to understanding the evolution of species. This knowledge helps the fight against disease, assists in the development of medications, predicting certain genetic diseases, and in discovering basic relationships among species in the history of life. Now, in this video, I will cover special protein databases. Let's we come on protein databases. First, Uniprot Kb, the Universal Protein Resource Knowledge Base, is the central hub for the collection of functional information on proteins. It consists of two sections, SwissProt and TrEMBL. SwissProt is an annotated protein sequence database established in 1986 and maintained since 1987 by the Department of Medical Biochemistry of the University of Geneva and the EMBL Data Library. The SwissProt protein sequence data bank consists of sequence entires, and sequence entires are composed of different line types, each with their own format. The SwissProt database distinguishes itself from other protein sequence databases by three distinct criteria: annotation, minimal redundancy, and integration with other databases. First, we understand what is annotation. In SwissProt, as in most other sequence databases, two classes of data can be distinguished: the core data and the annotation. For each sequence entry, the core data consists of the sequence data, the citation information, and the taxonomic data, while the annotation consists of a description of the following items: functions of the protein, post-translational modifications, for example, carbohydrates phosphorylation, acetylation, GPI anchor, etc. Third, domain and sites, for example, calcium binding regions, ATP binding sites, zinc fingers, homeobox, Kringel, etc. Fourth, secondary structures. Fifth, quaternary structures. Sixth, similarities to other proteins. Seventh, diseases associated with deficiency of the protein. And eighth, sequence conflicts, variants, etc. Second is minimal redundancy. Many sequence databases contain, for a given protein sequence, separate entires which correspond to different literature reports. In SwissProt, we try as much as possible to merge all these data so as to minimize the redundancy of the database. If conflicts exist between various sequencing reports, they are indicated in the feature table of the corresponding entry. The last one is integration with other databases. It is important to provide the users of biomolecular databases with a degree of integration between the three types of sequences related databases: nucleic acid sequences, protein sequences, and protein tertiary structures, as well as with specialized data collections. SwissProt is currently cross-referenced with 24 different databases. Cross-references are provided in the form of pointers to information related to SwissProt entries and found in data collections are other than SwissProt. Now we come on the second section of Uniprot, TREMBL, a computer annotated supplement to SwissProt. TRMBL, translation of EMBL nucleotide sequence databases in 1996. Why TRMBL? Increased data flow from genome projects to the sequence databases to maintain the high annotation quality to make sequences available as quickly as possible. TREMBL consists of computer annotated entries derived from the translation of all coding sequences in the nucleotide sequence databases except for coding sequences already included in the SwissProt. It also contains protein sequences extracted from the literature and protein sequences submitted directly by the user community. Let's come on the third type of protein database. ProSite is a protein database. It consists of entires describing the protein families, domains, and functional sites, as well as amino acid patterns and profiles in them. These are manually curated by a team of the Swiss Institute of Bioinformatics and tightly integrated to SwissProt protein annotation. ProSite was created in 1988 by Amos Bayrock, who directed the group for more than 20 years. ProSite's uses include identifying possible functions of newly discovered proteins and analysis of known proteins for previously undetermined activity. Properties from well-studied genes can be propagated to biologically related organisms, and for different or poorly known genes, biochemical functions can be predicted from similarities. ProSite offers tools for protein sequence analysis and motif detection. It is part of the XPacy proteomics analysis servers. The database ProRule builds on the domain descriptions of ProSite. It provides additional information about functionally or structurally critical amino acids. The rules contain information about biologically meaningful residues like active sites, substrate or cofactor binding sites, post-translational modification sites, or disulfide bonds to help function determination. These can automatically generate annotation based on ProSite motifs.